to demonstrate that light is necessary for photosynthesis. Requirements: potted plant, petri dish, boiling tube, alcohol 70%, iodine solution, Bunsen burner, forceps, beaker, water, dropper, black paper. Procedure: take a healthy potted plant and place it in dark for about 72 hours so that the leaves become free from starch. After 72 hours, select a leaf on the plant and cover a portion of it on both sides with black paper. Keep the potted plant with the covered leaf in sunlight for a few hours. After a few hours, detach the covered leaf from the plant and remove the black papers. Boil the leaf in water for a few minutes. Now boil the leaf in alcohol in a water bath till it becomes dull white due to the removal of chlorophyll. Wash the leaf with water and add iodine solution. Observation: the portion of leaf which was covered does not change color, whereas the portion which was uncovered turns blue-black. Inference and conclusion: the portion of leaf which was not covered received all the factors necessary for photosynthesis. Therefore, that part could only photosynthesize, whereas the one which was covered could not manufacture food. The presence of starch gave the positive test with iodine, that is, turned to blue-black color. Photosynthesis is the process of manufacturing of food by the green parts of the plant, especially the leaves, in the presence of sunlight and chlorophyll, with the help of carbon dioxide and water. To show that chlorophyll is necessary for photosynthesis. Materials required: coleus plant, watch glass, test tube, 2 beakers with water, iodine solution, alcohol, white paper, Bunsen burner, forceps, tripod stand with wire gauge, and dropper. Process: pluck a coleus leaf from plant. Coleus leaf contains both green and non-green areas. Transfer the leaf on a white sheet of paper and draw its outline. Mark the green and non-green areas carefully on it. Now boil this leaf in water to kill it. After boiling in water, again boil it in alcohol. The purpose of boiling leaf in alcohol is that it dissolves greenish part, that is, chlorophyll. Then wash the leaf in cold water and place it in a watch glass. Now pour some drops of iodine solution over it. Compare the leaf with the drawing you had made. To demonstrate that carbon dioxide is needed for photosynthesis. Requirements: potted plant, conical flask, clamp stands, sodium hydroxide solution, cotton wool, lime water, water, iodine solution, beaker, alcohol 70%, Bunsen burner, boiling tube, watch glass, forceps, dropper. Procedure: destarch the leaves of potted plant by placing it in dark for about 72 hours. After 72 hours, select 2 leaves of approximately the same size attached to the plant. Insert 1 leaf in a conical flask containing 0.4 microns sodium hydroxide. It will remove the carbon dioxide. A cotton wool plug soaked in lime water will prevent carbon dioxide from entering the flask. Set up the second leaf in the same way as the experimental leaf, but this time use water instead of the sodium hydroxide. Plug the flask with cotton wool soaked in water. Keep the setup in sunlight for a few hours. After a few hours, detach both the leaves and dip in boiling water for a minute. Now boil them in alcohol over a water bath till they decolorize. Wash them in water. Then test with iodine. Observation: the experimental leaf remains unchanged, that is, decolorized, while the control leaf becomes blue-black, showing the positive test with iodine. Inference and conclusion: the experimental leaf showed negative test with the iodine because in the absence of carbon dioxide, no photosynthesis took place. Hence, no starch is formed. While the control leaf showed positive test with iodine because due to the presence of carbon dioxide, starch was formed. It proves that carbon dioxide is a necessary factor for photosynthesis.